morning.
Oh, Waalaikumsalam, Prof. Good
Yeah,
morning.
sorry, I
Good
was
morning.
very tired. I reached last night from Alexandria and I
Yeah,
was sleeping today. Please, either Dr. Nazrin, greet him on my behalf,
yep,
and you or Dr. Lisa, or if you are with his CV and everything.
yes.
Uh, and then at the end, maybe I will able to join to say thank you.
All right. No worries, Prof. I believe we are actually live at the moment.
Okay,
Okay.
thank
All
you.
right. Okay, Prof.
Thank you very much.
No worries. Okay. Assalamualaikum and good morning, everyone. So, thank you very much for everyone who are alive with us today at the moment. Okay. Let's just give a few minutes to kind of settle down. Right. Uh, just give some time for people to start joining in. So we are live right now from ICA Facebook page. So ICA stands for Innovation Center in Agri Technology for Advanced Bioprocessing. Okay. So let's just give a couple more minutes. Okay, I can see some audience already start joining us live. Okay, good morning, everyone. It's 10 a.m. right now in Malaysia. I think some of you are joining us live from quite some distance in the globe. So thank you very much for spending your time to join us today. Um, I think maybe I can start with some introductions a little bit. Okay. So today we are having our second ICA webinar series on bioeconomy, food security, and healthy life. Okay. And the title of our topic for today is Utilizations of Non-Timber Forest Products for Health Purposes. Okay. So our topic will be delivered by our guest speaker, Dr. Rico Ramadhan, who is an assistant professor at Universitas Airlangga, Indonesia. Okay. And I will be helping out as a moderator. My name is Dr. Nazrin Aziz. I'm a research fellow for Innovation Center in Agri Technology at UTM Pago campus. And at the same time, I'm also a senior lecturer at Department of Bioscience at Faculty of Science, UTM. Right? And I think we can. I can start over with reading um, biography. All right. Okay. So let's open file. Okay. So we have over here Dr. Rico Ramadhan, who is an assistant professor in the Department of Chemistry at the Faculty of Science and Technology, Airlangga University in Surabaya, Indonesia. He is currently serving as the head of the division for exploration and synthesis of bioactive compounds at the University's Center of Excellence Research Center for Biomolecular Engineering. He is an accomplished organic chemist. Dr. Rico Ramadan specialized in forest product chemistry and bioassays. He has extensive experience in bioassay guided isolation of bioactive compounds and quantitative profiling of phytochemicals from medicinal plants and leveraging ethnopharmacological knowledge. His expertise includes studying enzyme inhibition activities of secondary metabolites such as alpha glucoside and tyrosinase, as well as exploring the kinetic mechanisms underlying these enzyme inhibitors. Additionally, he is skilled in the chromatographic separation of secondary metabolites and instrumental analysis using spectrometry. Um, okay, Dr. Rico Ramadan, how are you today?
Oh, I'm fine. Thank you, Dr. Nazrin.
Very good. Okay, thank you very much for taking your times and joining us, and actually for accepting our invitations to be featured as one of the speaker in our ICA webinar series. Thank you very much, Doctor.
Welcome.
Okay, let me just check again on our Facebook page. I think we have quite some audience at the moment. So, how are you, Dr. Rico? Do you think you want to start away with your presentations, or we want to 4 or 5 minutes, a little bit relax it?
Oh, tak apa, kita langsung saja. We
Okay,
go
all right.
straightforward.
Okay,
So we
all right.
can short the time for discussion, something like
Okay, very good. Okay, so ladies and gentlemen, present to you Dr. Rico Ramadan with his topic utilizations of non-timber forest products for health purposes. Dr. Rico, the virtual floor is yours.
Thank you very much, Dr. Nazrin. For us all, I would like to say thank you for your ITM team, Paco, and from your campus also. And I would like to say thank you to early morning to Professor Hisham El Hanshi for attend this event. So I would like to share about something that I would like to talk today. I hope all of you enjoy about the sharing knowledge. No need to be serious in this event. We try to, yeah. Okay, this I hope. Recognize it. This is my PPT is
Yeah, it's
well.
still, okay, all right, it's
Okay.
full view at the moment.
Okay. Thank you
good.
very much. So, for all of you, I would like to today share our knowledge in the title of non-timber forest product for health purposes. But actually, all the people, especially in Asian region, non-timber forest product sometimes like it's not familiar because many people talk about the medicinal plant, something like that. So actually, what is non-timber forest product? And today we would like to talk this in ICA Webinar Series 2 in aspect of bioeconomy, food security, and health life. So my name is Rico Ramadan, that Dr. Nazrin already introduced me, and we're going to our slide. Yes. Before that, I would like to introduce my University Center of Excellence, Research Center for Molecular Engineering. Actually, we are inside. We have 4 groups. The one is proteomics, and second one exploration and synthesis of bioactive compounds, and the third one applied biomechology and bioresearch technology is bioinformatics. All this research group, we are joining together to build University Center of Excellence. So our research center, we are integrated research group. So any problems we try to dissolve together to make any related knowledge. So this is our strongest research group. It's not only talk about not only talking about natural product, but we're talking any resources and related non-timber forest product and using the approach like bioinformatics. So what we do in exploration of synthetic or biotic compounds group? Actually, we also do synthesis like hydrolytic development methods, discovery non-timber forest product and based on ethnopharmacology, discoveries of biotic material for drug discovery, and also do derivation of natural bioactive compounds, and also exploration natural product for enzyme inhibitor. When you look the picture in the left side, right, you can look like natural product. They have discovery, biosynthesis, and biotechnology. We try to combine. So we're not talking about natural product. We're talking all resources that we can make it more valuable resource, something like that. So we try to make some little bit different. So when we're talking about tropical forest product, for example, like a zone in Malaysia and Indonesia, especially like in the Sarawak and Sabah, we call the tropical forest product. Many people talking about when you go to tropical forest product, we're just thinking about the timber. But when you're talking about the forest, we cannot only talking about the timber, but we have also non-timber forest product. What's that, right? Many people thinking like that is useful or not, something like that. Yeah, we can look in here in the picture. Non-timber forest product actually such as mushroom, honey, and then have some like bulb, have the resin, and also have the like a nuts, and also have medicinal plant. So when we're talking medicinal plant, actually there is the small part of non-timber forest product. So many people, when we're talking about medicinal plant, it means you are already talking just small part. But when we're talking about non-timber forest product, we can explore any as we can about what the non-timber forest can be help us, not only for economy, but also for health, something like that. So when we look this picture, we can have the some view, like, oh, so it means non-timber product is more useful for example, like for health, for any drug discovery, something like that. Rather than just talking about the timber, right? Many people know this is timber for example, for just furniture or something like that. So the next one, I would like to talk how we are using the approach to get the knowledge is very easy one to support our research and our development products. For example, when we talk about the some area, we're talking about the local people. For example, Malaysia also, Indonesia also, Sarawak, Sabah, and also the people like in the we call, I remember Malaysian people called suku asli, right? So they have some knowledge that we never explore before. Sometimes they're afraid, but we should like slowly talking with them, discuss with them. And actually around the world, they have shown any ethnopharmacology that sometimes we thinking they just do like a ceremonial, but actually inside we can get some scientific report. This ethnopharmacological information from local people is very important to develop our research. For example, one of my project now in the central of Kalimantan. This is the border of Sarawak and Sabah. I using animal behavior because Borneo is a huge home of orang utan. When orang utan using the plant, we try to study, right? When we study, and also the local people, for example, like Dayak and any people that live in around the forest, we try to explore what they have. We come to their ceremonial, for example, like this picture, right? This picture was taken in the deep of Kalimantan. So we try to ask what the plant, what source they use for this one, because this is the ceremony. Sometimes the local people they use for healing, right? So we try to collect the information. This is very important that I ever study about ethnopharmacological information. So what is ethnopharmacology, right? Ethnopharmacology is interdisciplinary and multidisciplinary approach in drug discovery that involving description biological activity, investigation for drug remedies. The study is the how, right? The how to produce natural product for therapy. So for example, this is the map of Borneo Island. They have part of Indonesia, Sarawak and Sabah, also Brunei. You know, I ever study in the border of Sarawak and West Kalimantan. You know, the plan, Sarawak also have part of Kalimantan Barat, and Kalimantan Barat also have half of Sarawak. This is very important when you study. For example, I cannot go to Sarawak because I cannot take some material, right? So I can study in the border. Many people stay, local people in Kalimantan in the border, they use plant for example, for economic, from food, for cosmetic, and also for health healing. So this information is very important. So I'm not going to Sarawak to get some plant because the ecosystem is quite same, right? This is the important when we are still should we have collaboration. For example, just in this area, something like that. In the daytime, I can some like collect some plant that also have in Sarawak. For example, longifolia. The name is in the Kalimantan Barat, di Tekiriho, but actually this one is pasak bumi or tongkat ali. Tongkat ali a lot in Sarawak and also a lot in Kalimantan. So we can use together, right? So this important thing is the local people can give information. Tongkat ali or pasak bumi is not only for example, for the man, but actually also for drugs, for example, like that, for also for healing and wound healing. And also, five this plant that I collect is also from the border of Malaysia and Indonesia. For example, like ICA this one is very famous in Borneo. This is we call ironwood. The people in here they build their home using this plant. Very strong because this plant is contain a lot of polyphenol. Very strong. When they are contact with water, I mean when you soak in water, the wood is more very strong because the binding of the polyphenol is more and more and more. Something like that. This we call the ironwood. It's very important in Sarawak, Sabah, and Borneo. And then we know about the ethnopharmacological knowledge from the local people around this border. Later I would like to tell you how important ethnopharmacological. So how ethnopharmacological is going on, right? This is actually intergeneration transfer by intertraditional medicine knowledge. So for example, me, now I hold some book from my grandfather, my grandfather, my grandfather. So I keep it. I have around 1,000 plant that I know my grandfather and give me some information. So when we know generation by generation, so this is can make to bioprospecting natural product. And then what the content? The first is knowledge. Knowledge is transferred between individual. This knowledge is applied by traditional healer. So we no need to uh, take directly random, right? We already know information. For example, like Tongkat Ali or Pasak Bumi. Many people long time ago used that for the fertility for men. But actually, in another place in Kalimantan, they also for wound healing. So if we combine this information, our research, for example, when we do research about this plant, is more comprehensive research. So, and then the second one is the result achieved through administration therapy across generation by generation. And we know safely or not, we no need go to the test for the safely or for some disease toxicity or not, something like that, because our old generation already use. This is very important for research. When you deal about, for example, like halal or this compound or this plant is toxic or not toxic, we can use this information. And ultimately, the iterative process is driven identification the most effective natural product to treat the specific disorder. For example, everyone know, for example, like piperine. in Kalimantan, they have many piperin. For example, the green one, the red one. But when I collect information in Sabah, right, many people have bleeding from the nose. And in Sabah people, they're using how to stop the bleeding from the nose. They're using the piperin. This information can make some inspiration to test this plant, I mean piperin, to anti-inflammation and for for example, for anti-bleeding, something like that. This is the inspiration that I also use how to deal with the medicinal plant. And then how to discover it by active compound. For example, many researchers are thinking, oh, we just go to the market. No, actually, when you stay in the forest, you can have the forest product and using local people, and you just take it some part of non-timber forest product. For example, medicinal plant, only medicinal plant. And then you try to chemical screening and bioactivity and using the modern approach, for example, using LC-RMS, they have database, AOC, GCMS. You can hit the target right very quickly, very quickly. And then, for example, you have some plant that rare. For example, local people said, oh, in the plant, you collaboration with biology, and then have you know the potency from the local people, and you directly do something like this. You can do press report for the rare people that I always do. And then this also actually the key how to increase citation, because when you do and then you the first report, who is the first report of the plan, the many people will follow you and they will cite your paper, and then you should do as many as comprehensive, something like that. If I didn't do isolation, I do just like BioSA like this and do LCRMS and try to do for profiling, something like that. And then this is that I have done many steps how to exploration secondary metabolite in Indonesia flora. For example, I take some medicinal plant in East Kalimantan. This is the how to do, yeah. For example, you do crude extract and then you do screening all as and you have try to fractionation and try again which one active fraction and bioassay guidance extraction, so you can know which extract that you will take it without any random, right? After you know, for example, after you know the semipolar, the good one, the active one, you can go further for chromatography. After that, you do the fraction and you do again for assay. In the fraction, you check again assay, and then you do the NMI, SPLC, and LC, and any uh, any method that you can provide in your lab, and then you can do again for analysis. For example, for me, I just focus in diabetic complication. I try to use the diabetic approach and also using the reactive oxygen species representative of free radicals, and also do kinetics mechanism, something like that. And this is the one success that I have done for my research. I tried to exploration some compound after isolation, and then I just know this compound is can good in free radical scavenging ability in antioxidant ABTS, DPPS, or nitric oxide. Sometimes some compound good in ABTS and DPPS, but not good in nitric oxide. But don't worry, we still have another assay that we should think. For example, like this compound, this is the acylphenoldimer that I got from the Myristica. And I think in Malaysia a lot, Myristica, Palak, something like that. This compound can good in nitric oxide, good in antioxidant ABTS and DPPH, and also good for enzymatic in alpha-glucosidase, in bakery yeast and red intestinal alpha-glucosidase, in maltase and sucrase. And then not from that only, I also do something is more go further, because I get a lot of compound in dimer, and actually this is acylphenol with the hydroxy attached around in aromatic rings. This is quite important and quite interesting also. So I do DNA damage protection using some plasmid and using the three plasmid and try to use Fenton reaction to break their bonding in here. And then I put my sample and I try to check how far this Fenton reaction can break this binding from the plasmid. So I know the plan of from the structure, I can know that my structure can protect DNA even from the closed circular supercoil to open circular and linear. At least my compound didn't disappear the DNA. This means my compound can protect the plasmid rather than they are break all and from electrophoresis we cannot see anything. That is indicate my plasmid is broke. It means my compound cannot protect. So I just using the simple reaction because I'm not focused too deep in biochemistry, but I try to know because to going further drug discovery in Indonesia is very long because we still should be approved by preclinical clinical tests, something like that. But at least our plan is promising to develop more deep, something like that. And another one that I also success, yeah, this is information this plant also grow in Sarawak and Sabah. I got this one from ethnopharmacology, something like that. And another one is from Garcinia. I think in Malaysia, a lot of Garcinia. And from Garcinia, I take the fruit because in this plant, fruit is very rare to get. This fruit is quite different with Garcinia mangostana. This one is Garcinia parvifolia. And I got Garcicinidon A. This structure is actually Depsidon. And then we can look in here, right? Very promising because they also have the non-polar part. This means they have granulated, right? And instead of the phenol group, something like that. And I also using ethnopharmacology. Why? Before I took this plant, I have some interview with local people, especially Dayak people, that they have a lot of some information about this plant because they use this fruit for cooking instead for recipe this fruit. Because this fruit is have the flavor sour. After that, I just thinking if sour is mean this compound is quite interesting based on the biosynthesis in natural product. So after that, I tried using the chromatography technique and using this approach, and finally I got this structure. And finally, I tried to test this structure to the free radical scavenging activity. Again, I using DPPS, ABPS, and ABTS, and also nitrate oxide. And also using the approach of alpha glucosidase, using the yeast as the screening. And I also do red intestinal alpha glucosidase using the substrate maltase and sucrase. Why I using this one? Because in our intestine, we still made of the maltase and sucrase when we eat something like a polysaccharide, something like that. So this is how to deal with the natural product, especially non-timber product. Not only talking about the stem or maybe the wood, but we're talking about the fruit, how they are useful for health. And the next is I talking about what the next. After we talking about compound, we isolate based on ethnopharmacology, and then what we next, right? We try to do modification by reaction to know how important hydroxy in this some particular compound, right? So we try to change the hydroxy in here to be methoxy, and then how to effective hydroxy when they replace to using the some methoxy, something like that. So we can look, right? This is compound one, is the original one, have the R2OH in here and have H and EH. I mean they have 3OH. You see, number one is more effective than number 1A after modification. This means the hydroxy in here important for this plant to antioxidant and for diabetic. We can look in here, right? Number one is more effective IC50, lowest than others. right? It means the IC50, the lowest ST50 is mean the highest anti-diabetic. I mean the good one. We can look after I do modification, try to replace OH in this position and in this position, OH in this position, I change to methylation reaction. So what happened? You see, they are gradually and try to increase. I mean, the growth is increased, but the activity is may not increase. This is how to prove hydroxy group in our compound is important. So I try after that what I doing. I doing the kinetics by linear plots to know how the mechanism of my compound, right? The compound one is they have the compound one, they have the mechanism. We know, right? In enzyme they have the competitive, non-competitive, mixed, something like that. So after that, we know our compound they have non uncompetitive and mixed competitive and also mixed inhibition by using the enzyme. So we can know my compound is good for activity and also can know the mechanism after that. So we can prove uh, step by step our compound, something like that. Not only that one, I also try to do many as isolation from Myristica species and also get new compound. This is hospital A. I got the new compound. They have like a OH in the meta position and also have hydroxy can attach in here and also have secondary alcohol in here. This is very, very interesting because I got, because I got the three new compound from Myristica. And then I try to check the mechanism and also IC50. And then also for antioxidant, you can look in here, right? Almost the compound that I isolate expect the number 3 because they have the glucoside in here, active against alpha glucosidase and very also active against free radical. This means this compound is have dual function, not only for anti-diabetic in the screening, but also for antioxidant, something like that. And then go further, I try to do some assay using the maltose and sucrose, and then we shoot kinetics, and then we know the mechanism is mixed inhibition, something like that. And another one is another compound that I success to isolate from Indonesian flora is some chalcone, yeah, 2.4 hydroxy chalcone, and also some dimer, myristicin G and myristicin D. And then we go further also for anti-diabetic and antioxidant. And then we also do kinetics. This is very important to know how their mechanism and also to check how the effective IC50 of compound that we already isolate. Another one also I have done in exploration of secondary metabolite is how to isolate from the Charioptagal. I think everyone know Charioptagal is family of mangrove, right? But how to deal with mangrove, this is very important because they have a lot of resin, right? So yeah, this compound is not new, it's very common, right? Like lupiol, bitulon, and betulin. I isolate from Charioptagal leaves, and then I got the major one, and then try to test the alpha glucosidase, and then compare to acarbose, right? You can look in here. Even 3 of them still active. I mean, we compare to acarbose, they still have activity for inhibit alpha glucosidase. And then I try to do the mechanism, right? To do the mechanism, and then how uh, the mechanism that they have to inhibit the alpha glucosidase. And then we can show in here that the compound lupiol, bitulon, and betulin, actually they can inhibit in the uncompetitive, something like that. So yeah, when we study about the natural product, uh, not me, not only talking about natural product, but also biotechnology. So I try to using approach related to enzymatic also, or radical also, and then how to relate the chemical reaction that I cannot do in vivo, but I can try to using in vitro that approach to in vivo, something like that. For example, sometimes nitric oxide you can do by some macro pack, right? But in my lab, I cannot do that one. I should develop some methods or maybe just look around what people have done with the nitric oxide by chemical reaction, something like that. So we can do some like preliminary and then we can know. Even sometimes in vivo and in vitro, sometimes we cannot like keep it. Just like if in vitro is good, it's not guaranteed in vivo good, something like that. So, but just we try. We have the preliminary data, we have the isolation, we have data to make like a proof that, oh, we can isolate the compound using several techniques, and then we try to do in vitro, and then we know how deal with that compound, something like that. Sometimes many students and also my students also sometimes we cannot deal some isolation how to the effective method. This is also I try to do some shortcut research to make research is not to take long, something like that. And then yeah, summary, when you have the ethnopharmacological and then you're just thinking how to get the good one result, right? Many people want to get the new compound. Many people, when you deal with the natural product, many people want to have a new compound, right? But for me, in my research group, we're just talking about how the good bioactive compound with any bioassay activity that they can cover. Because when to get a new compounds, sometimes we need to how to say it, like a thinking many possibility technique, because sometimes new compound is the very tiny amount, right? So what we should do is mean the observation, identification, and description, also experimental investigation of ingredient, of the effect ingredient and the effect such ingredient drug is truly interdisciplinary field of the research, which is very important in study of traditional medicine. So we cannot only just take a plane, go to the jungle, go to the forest, without any observation, without any identification, and try to describe for example, like chemists, we also use chemotaxonomy. This means you try to look the family, what the family of the plant that people already have done, and then what's compound kind of the what the type of the compound, something like that. So this is the how to get the high hit target result in natural product and also traditional medicine, something like that. So yeah, when we know about the biological diversity, we have human knowledge and practice, and we try to looking by lens of ethnobotany, so we can get good result in a non-timber forest product, not only medicinal plant, honey, something like that, and also resin, and also mushroom. Like now in my project, I just have done to do isolation in propolis from the stingless bee, and we got some compound. It's very interesting in anti-cancer. So, and then actually when you do with the stingless bee and like honey and some we call, I think in Malaysia also called kelulut, we actually support the conservation. Why? Because when we get the sample from the honey or propolis, we didn't cut the tree to get sample for the research, something like that. So this approach sometimes I use for for closely to get the research is still conserve the plant, something like that. Yeah, this is that I with my collaborator, right? My collaborator that we have done that I told you. Okay, police. Yeah, I collaboration with my former professor in my former university from Universitas Warman in Borneo in Kalimantan, and we're talking about stingless bee. How stingless bee can useful for health purposes, something like that. We can know, right? Some people didn't thinking of not only medicinal plants, but also non-timber products, for example, like propolis. This paper we're going to study from 2021 going to 22 because have the COVID-19. So we try to how to screening stimulus B for inhibition AAC2 spike protein binding, something that we do collaboration with the Kyushu University, and we publish in Malaysian Journal of International Science. And also we're going to further isolation, right? And we need to know about the effect of remunerative flavonoid that have in product of propolis. And also the last one in 2024, we also evaluation product propolis for cytotoxic activity in many cells, something like that. So when you do research, you should thinking how to sustainable the research, not only for example take one and finish in the 1 year, but not have continue something like that. I hope this can make inspiration. After that, we can thinking after this one. It means the propolis actually is exactly have the potential for health purposes, something like that. And this is also that I have done for analysis any non-tuberculous product and make a book, some book in the Springer and try to use whitening agent. And I'm surprised because some non-tuberculous product also good for whitening, especially in anti-melanogenesis and also tyrosinase in L-DOPA in vitro using L-DOPA substrate. And also using the GCNS analysis to know the Diospyros species, we call in Indonesia ebony, also help can use for inhibit alpha glucosidase and PrEP activity. This is related to actually PrEP activity is related to the iron and also can reducing agent that related to antioxidant. And we also do phytochemical analysis conjunction with in vitro glucosidase, something like that, and any plant, and we can make it database which plant is good for alpha glucosidase and good for like antioxidant and good for whitening agent. This is very important result, right? Because we can know everything, not only talking about isolation, but actually in suppression and extract also is good, something like that. And this is the one of our successful group that we are recognized by government of Rico City. Our research project is the government use our research project and involved in this project. Major of Balikpapan, we try to the identification the flora that now the icon of Balikpapan City is endemic flora. This plant is the member of Sigiberseae. Now this plant is protected by government because our result from research and collaboration with one suggest to have some government policy, something like that. And another one, this is the international research consortium that I try to build with the Malaya University and Chulalongkorn University. We're talking exploration by active compound from Dipterocarpaceae, because Dipterocarpaceae now is very huge in Kalimantan, in Borneo also, Sarawak, Sabah, and I also got some information have some Dipterocarpaceae in the around the Semenanjung. They call it Dipterocarpaceae johornensis. That is very important also because many people said in ethnopharmacology, Dipterocarpaceae good for wound healing, antibacterial, and anti-cancer because they a lot of use polyphenol. I think in the Semenanjung they have like this plant. Its name is Dipterocarpaceae johornensis. If I'm not mistaken, they have name Johor. And also, yeah, with Dr. Nazrin, actually, we have some matching fund, UNE and UITM with Dr. Nazrin. We are close research. Now we are trying to identify each other, help each other to some exploration and try to unlocking something between Malaysia, UTM and UNE. And we try to make a more valuable, not only 1 year, but we try to make like a 5 year later. So it's make some like a consortium, and our result, our collaboration is more strong and strong, and it's like collaboration not only how to in research, but also, for example, like a student and many activity, for example, like with Dr. Nazrin, and also with my colleagues from Thammasat University, we try to make it Airlangga Research Fellowship in 2025. Maybe after matching fund with Dr. Nazrin, I will ask Dr. Nazrin also have the research fellowship UTM at Airlangga. This is more make strong collaboration, not only 1 year, but I try to make some research can sustainable until 10 years and make it some product, and yeah, possibility can we make I think like some activity that make more strong collaboration. And yeah, this is one of my postdoctoral researcher from Chulalongkorn University. I also welcome to any participant attending today when you ask about my research group. We open for any possibility research, something like, because my university now is very open for joint collaboration, joint research, joint publication, and joint advisory. Also, any activity can support each other is make more valuable. We open it. Okay, maybe that one I can share for all of you guys. If any question in this, if the time is not enough, you can visit the Airlangga University in this website. I mean, in University Bioeconomy, you can find me in there. Thank you very much for your attention, guys. So I hope I will send back to Dr. Nazrin.
All right. Thank you very much, Dr. Rico Ramadhan, for that very interesting and inspiring research on non-timber products. Okay. So I think let's just go back to Facebook just to check first. Okay. So we have some quite numbers of attendants listening to your presentation just now, Dr. Rico. Some of them were quite excited with what you have done earlier. If I may, we can go into Q&A and short discussion session.
Yes.
At the moment, I would like to have the honor of asking the first questions actually. As Dr. Rico said that we have been working for quite some time, right? So I was wondering, because when I look at one of the results you showed, one of the compounds that you have isolated has the ability to protect DNA molecules from degradation. So me myself, when I look at that as a molecular biotechnologist, it really got me excited. So what's next, Dr. Rico, from that part of research? So how do you intend to further uh, deep dive the research into that aspect?
Yeah. Okay. I directly to answer, Doctor.
Yeah.
Okay. So yeah, the first I thinking the structure, Doctor. Why I can directly to the DNA damage protection. The first thing is we can know, right, the plasmid, or not only the polar compound, right, but also they have like some membrane, like from the maybe some membrane is not like not cannot protect by the polar compound. So when we look my compound is have the long chain. Long chain is quite non-polar. So after that, I read some paper. This compound can protect some, I call like a plasmid is representative of DNA, right? I mean, I cannot go
Yeah.
to DNA directly. I'm using this plasmid. So
Yeah.
I read some paper. I just want to try, is this possible or not? My compound, they have long chain and have phenol, can protect DNA by the Fenton reaction. Because when Fenton and my compound, they can protect. This is the principle like antioxidant.
Mm.
But how if the Fenton I break by DNA I break by Fenton, can or not if I protect first DNA and then Fenton can attack DNA or not, something like that. Just the impression like that. And I read some paper from ACS USA. They said, yeah, the compound, the type like this can protect some molecule, for example, DNA or protein. And I
Mm.
tried with. like that, doctor. Because I do the simple, just electrophoresis. I'm not going further because I don't know expert go do further something like that, doctor. This is inspiration for me. I look the compound first, the structure.
I see. Very interesting. So maybe I or maybe one of the audience who are from molecular biotechnology background can collaborate with Dr. Rico to further studies
Yeah.
about this aspect. All right.
Yes.
Okay. So I think I have I'm looking at some questions coming from our audience. So we have the first questions from Dr. Siti Nur Azina Rashid, is ethnopharmacology still being done nowadays by the native people? What do you
Yes.
know about it, Dr. Rico?
Yes. Ethnopharmacology is still being done today. Sometimes I try to give you some secret, right? Sometimes the academia they don't want to go to the forest deep one.
Mm,
You just do chemotaxonomy, and then sometimes the chemotaxonomy is mean many people already done, right?
yeah,
But when you go to like border of Sarawak, border of Sabah,
yeah.
something like that, we can look. Many people still the ethnopharmacology, how they are reach the hospital too far. They're using medicinal plant to protect themselves. I have
Mm,
some experience in border of Sarawak. Some Dayak people, they have a bite with the knife and they have
mm,
them scar. And finally, they take it some plant and using the water and they put the plant go to the wound.
mm,
And what happened that one? They said, oh, it's quite what how to say the wound. They said waiting for a week something. And then I
mm,
look after that when I came in there, and then I look. I said, yeah, the wound is look like the fibroblast is look like come back to normal.
mm.
Still have
Okay.
scar, but I didn't look the red one. It means the red one is inflammation, right?
Mm,
It means this plant is not only for wound healing, but also can reduce the anti-inflammation, right?
mm.
Uh, this inspiration, sometimes we should go to the field. We cannot
Yeah.
go to the some area only, but we just like stay in the 1 week talking with them
Mm,
and writing some note what they're doing, something like that, Dr. Nazrin. So yeah, still this is still have in native people, yes, especially the people that stay far from the city. For
yeah.
example, like in the Semenanjung, they have suku asli, right?
Yeah. Yeah.
Semenanjung suku asli. Have the some people do ethnopharmacology like talking, like look like a discussion or interview that
Yeah.
people
Yeah.
is very important,
Mm,
very
yep.
important because
Agree.
sometimes they didn't believe us,
Mm,
right? They didn't believe us. So you should slowly. For me, when I go to the jungle, I'm stay 2 weeks and talking
wow.
with them, talking with Kepala Kampung, talking with the head of the village, something like that.
Mm,
And then we writing. And the lucky one, when you go to the some village, when they have the ceremonial, it's very lucky because all the all the recipe they open because they do the healing, right? The ceremony of healing. Yeah, something like that. And then I writing the plan. And
mm.
then now today we can use the picture from Google, right?
Yeah.
For example, they have plan and then we just take picture of this plan. Oh, we can using this
Oh,
information to the family or cat species, something like that.
identify the plant species.
Yes,
Google
yes,
Lens. Yeah.
yes. That is very important for us, something like that.
Yeah.
And yeah, I think that one. If ethnopharmacology still have today, yes. Then I until today I still use for
Mm,
support my research, something like that, doctor.
yep. So in Malaysia, I think there are, I think, quite some numbers of researchers who are very active in documenting and,
Yes.
uh, as you said, interviewing our um, suku asli,
Mm.
indigenous people, to document their ethnopharmacological knowledge with the local herbs and medicines. So there are some efforts going on over there. But as Dr. Rico highlighted, what's important is to once after documented, then we have to scientifically help to prove it, prove the ethnopharmacological practice identify what is exactly the specific bioactive compounds that give it that special property. Okay. So we have our next questions from Associate Professor Dr. Noraza Basar. How can we ensure that standardization is maintained when scaling up, especially for extracts with commercial potential? I've heard about this, not to say problem, but maybe issue with
Mm.
natural product extracts is always about
Yes.
standardization. So, what's your opinion about this, Dr. Rico?
Yes, yeah, it is also a issue in Indonesia when we're talking about natural product, especially when you're going to Indonesia, we have jamu, right?
Mm, yeah.
The commercialization extract is mean you should like a step by step look the product that when you want to use for some product, right? Because when we're talking about the standardization, it mean from the leaf, for example, leaves from jamu. The leaf should be have not have mushroom or have a fungi, and then how when you take it is mean don't have something like how to say like should be clean, and
Mm,
then how when you take it in the rainy or not rainy, this is also have the standardization.
mm,
The question is been how to if this come on commercial potential. Yeah, this is have some barrier, researcher and industry, right? Sometimes industry need the validation, how to say like the product before going to be like some commercial product. This is the problem. But in my opinion, yeah, to be research like this, you should use the plant that is biomass. We cannot use the plant the rare one because impossible. You produce the biomass. You didn't use the biomass to be the product commercial without any how to sustainability, right?
mm,
Same also this question, such standardization. When they scale up, the important thing is mean you should have the biomass as the product before going to product because I'm afraid when they are mass production and when they cycle, we didn't have the raw material. This is a problem, right?
yeah,
Something that.
yeah.
So actually, in natural product, is mean we talking a basic one. But when we do some, for example, like kunyit, right? I agree this statement like kunyit can scaling up, yes, because it's biomass, right? Everyone can culture and either some land, something like that in 1 hectare or 2 hectares, something like that. But what happened if my plant, for example, I show you in PPT, I also do ginger
Mm,
that already protected by government Balikpapan. We cannot do anything. They just
mm,
protect first and research by tissue culture. This is can or not do tissue culture using the like can make it more and easy
mm,
to. If not, we cannot go into the scaling up something like that, doctor. So yeah, we should look this is biomass or not. How to
mm.
commercial the potential something like that.
Okay, all right. So, what if I understand correctly, in order to maintain that standardization when scaling up, first, we have to ensure that we have an ample supply of that biomass, and
Mm.
that supply must be of consistent quality. It must
Quality,
be
yes,
harvested at the same condition and at the same time to ensure that uh, the profiles of the compounds inside are roughly similar. Is that correct, Dr. Rico?
yes, correct.
Okay. All right. Good. Okay. Because I'm not a biochemist, so sometimes a little
Yes,
bit difficult for me to understand. Yeah. But I'm
yes.
learning. I'm learning as well. Okay. So next we have from Ms. Noraina Atira. How Dr. Rico foresee the role of advanced techniques such as bioinformatics or applied microbiology evolving in the study of bioactive compounds to validate the traditional uses of medicinal plants based on the ethnopharmacological knowledge. So, what's your opinion about using other aspects of not just the biochemistry sides, but we incorporate bioinformatics, applied microbiology, or maybe even plant tissue culture or mammalian cell culture study? Dr. Rico?
Yeah. This is a good question because my research, my center of excellence now, they do this one.
Okay.
I will answer one. For example, Dr. Nur Aina Atira said how to advance bioinformatics, right? In
Yeah.
bioinformatics, sometimes they can predict, right, the compound. For example,
Mm.
like that by using database, right, from database. And for example, we're lucky if we already know the species of the plant, so we can use the bioinformatics to predict our plant have a lot of the compound of this one or not, because the similarity of species or genus is quite similar.
Mm.
For example, uh, kunyit, a lot of like lot of compound with the type of phenol, right?
Yeah.
So bioinformatics try to use the approach to, for example, you choose for anti-HIV, for example, and try to use protein in database and using the precursor, using the SIV from protein from SIV and try to connect with this plant. This is have a binding or not before we going to isolation, right? So,
Mm.
for example, bioinformatics can give the some predict is very nice because we can go further. Can go further that I do isolation this one and try to quantification how many how many milligram from 1 gram. For example, something that bioinformatics can like make a prediction which compound can active or not active in some disease based
Mm.
on the protein, something like that, doctor. And in
Mm.
my research group, also I do done with that. For example, to prove that is true or not in ethnopharmacology, like kunyit or halia, in Malaysia sebut halia, is good
Mm.
or not for the coke, right?
Uh,
So the coke, what happened with the coke? And then we're looking for bioinformatics. What's protein can responsible for the coke or from for antibacteria or something? And then we can look what happened if the compound from halia we connect and binding with the in system of bioinformatics, and then which can binding and which one can inhibit. They can bioinformatics can predict, doctor, very
mm.
quickly, something like that. So
That's
that's why
nice.
many people know do profiling, for example, using LCRMS, and they know have many compound, and they just do send to bioinformatics, and they will
Mm.
do screening which compound is effective, for example, for HIV, because
Mm.
they based on protein, Dr. Nazrin, and then do for antibacteria.
Mm.
And then like me, now I do anti-malaria by informatics, something like that, to predict is true or not before I'm going to isolation. Something like that.
Mm. All right.
And yeah, and another one, applied microbiology. My college also do endophytic fungi. I think many people is very common fungi, right?
Yeah.
And one successful in my research center is mean the plant that I isolate, I got the same with the bacteria that I isolate, the compound.
Mm.
It means the endophytic fungi, sometimes they are have responsible the plant how to produce the metabolite secundary, something like that.
Mm.
Secondary metabolite sometimes quite similar. Sometimes we
Yeah.
don't know how the mechanism, but applied microbiology help us to know which bacteria can can like changing by transformation, maybe I don't know, because magnesia, and then the same plant at the same secondary metabolite that I isolate in the same plant, something like that. For
Mm.
example, this is plant leaf.
Yeah.
The microbiology just takes some bacteria around in here
Yeah.
and they culture, right? And then
Yeah.
they said, yeah, the plant is quite the secondary metabolite is quite same.
Mm.
And also do in my research center, yeah, they do also both in bioinformatics and also for applied microbiology. And that I show you in my PPT in the first, we have the research group by bioinformatics research group and applied microbiology,
Mm.
something like that.
I guess that's quite interesting because it's almost to say that the profile of the compounds that are
Mhm,
being produced by plants can be
mhm.
affected by the communities of microbes,
Yes,
like
yes,
the bacteria or fungi that
yeah,
grows around it
yeah.
or inside of the plant itself. Oh, that's
Mm.
very interesting. And
Yes,
just
doctor.
a follow-up questions on the bioinformatics usage. So does that mean that we must have the whole genome sequence of that
Yes.
plant in order to
Mm-hmm.
use to predict
Mm-hmm. Mm-hmm.
the
And
kinds
then
of compounds that they can
yes,
produce?
and
Oh,
also the
okay.
compound also. And what's bioactivity that related to protein? For example, HIV, anti-malaria. So we can look database in database protein bank,
Mm,
and with the protein that related to, for example, anti-malaria. So we take it that protein to be some model, something like that, doctor.
I see. Okay, all
Okay.
right. So moving on to
I
yeah,
can give us some information, doctor. For applied microbiology,
yeah.
maybe Dr. Nuraina Atira can look my paper, the new one,
Mm.
just accepted last week,
Ah,
talking
very
about
fresh.
the update, yeah, applied microbiology of endophyte fungi from Halia,
Ah,
something like
okay.
that.
All right,
Yeah,
very
talking
interesting.
that.
Do
Yes.
send me a copy of that as well, Dr.
Yeah,
Rico.
okay, Dr.
Okay, all
Nazrin, I
right.
will.
Okay, thanks. Okay, I'm getting the paper directly from the author of the paper itself. All right. So next, we have another questions from Dr. Azlina. Given the advancement in analyzing these plant samples, what percentage of them have actually been commercialized for health purposes, and are there specific factors limiting this commercialization? If I can just elaborate a little bit this question. So because I thought about this a lot quite sometimes. We have a lot of scientists and researchers working on the bioactive compounds of medicinal plants, and many times the researchers, they have successfully shown and identified which of the compounds are responsible for a specific properties. But then, like, what's next? What are the factors that limit the next step, which is
Mm.
commercialization?
Okay, doctor. Uh, I don't know the rule in the Ministry of Health in Indonesia. We have three. We
Okay.
have three type: jamu, and then Phytopharmaca is the very highest one, and then obat herbal terstandar. I mean the standard herb drugs. Indonesia have three type.
Okay.
So according this question, how the percentage them have actually commercial? Yeah, the first thing is should be biomass. So that's why product of Halia in Indonesia is a lot because before we going to produce, we sometimes the some entrepreneur they provide the some land, for example, 2 hectares to produce the biomass because when the production is come, so it's mean the sustainability, the raw material should be proven, right? Cannot stop because raw material didn't have something like that. So
Yeah.
this is the limitation. Sometimes the local entrepreneur, they just provide the small land. They sometimes cannot.
Mhm.
Another one is what the specific factor limiting this conversation. Sometimes is permit, and the
Okay.
second one is quality,
All right.
and then for example like consistent of the quality. For
Mm.
example, what happened if the rain come, what happened with product the essential oil is quite same or not.
Mm.
This is also should we have the treatment how to the commercialization. Sometimes only the people they have the huge the company can treat this one, right? But for the
Mhm.
local, it's sometimes the factor limitation. And also if not biomass and not edible, sometimes we are researcher have some barrier. It means your compound is your sample is toxic or not toxic. This is the question, right? Before we going further to be product, especially the product for food, something like that, right?
Yes.
Like Halia, many people didn't ask again, this Halia is toxic or not because traditionally many people use, right?
Mm.
Something like that, Dr. Najin. So actually, when we do commercialization, avoid some plant is not common, something like that.
Mm.
But we try to derivatization the product. For example, Halia in Indonesia now also have candy, gula-gula. Ah,
Okay.
yeah, right? Yeah, gula-gula from Halia. And then also, how to say it, like gula-gula rubber, but
Mm-hmm.
the flavor is Halia. Ah,
Oh,
yeah,
okay.
only
I
do
cannot
that.
imagine that.
Yeah, yeah. So we just do like that. We cannot do like any plan that even they good for drug discovery. Because, for example, I have some experience, doctor. My compound is good, or my compound is very nice for any assay. But for the clinical, my compound is toxic.
Oh,
Finally, all the assay is cannot go further,
I see.
right? Yeah. So what we should do? Yeah, the good one is make a jamu.
Mm,
Because yeah, jamu is traditionally is used, right?
mm,
But it's promising, yes, promising. But we should thinking biomass is very
mm,
important to be
mm.
get the raw material
All
in
right.
industry, something like
Mm,
that, doctor.
okay. So when we talks about commercialization, again, it goes back to consistent supply of
Uh,
biomass and
mm,
also the quality, a consistent quality for
yes,
standardization. It goes
yes,
back to Associate Prof. Dr. Norzabata questions earlier.
yes, yes,
Okay.
doctor.
All right. Yeah, I think that's one of the most common problems when we talk
Yes,
about natural products from
yes,
herbal and medicinal plants, which is consistent supply
yes,
of quality materials. All
yes,
right, we still have some more questions. So we have from Mir Mashudin. Dr. Rico, can you share any experience on extracting the phytochemicals through solventless technique?
yes.
Not without using any solvent, Dr. Rico.
Open, yes, yeah. Yeah, yes, this is very important and interesting. Actually, uh, many people using the microwave
Mm,
extraction.
okay,
Yes, yeah. In the my laboratory, sometimes because this equipment they're using microwave to extraction without any solvent because they're using the microwave, right?
just
To
normal microwave.
no, doctor, they have
Okay.
a laboratory microwave.
Oh, okay.
This is
I have
actually
a
don't have,
microwave
yeah,
at home, so
yeah.
it's a different types.
And
Okay.
and also the second one is using the the assisted by ultrasonification,
Okay.
doctor. Yeah,
Ultrasonication.
both of this one is as long as I use both of this one is very effective because ultrasonification can break the cell, so
Mm,
our metabolism powder can go out from the cell, something like that,
okay. All
without
right.
any solvent, something like that.
Okay. All right.
Yeah, and
And
microwave also.
for that ultrasonication, I assume your plant materials are immersed in just water, normal water.
Yes, something like that.
Okay.
The cheap solvent, not organic solvent, the cheap solvent,
Okay. Okay. All right.
something like that.
Cool.
For essential oil,
Mm,
I do the three method. The first steam, and then boil, and then using the microwave, something like that.
mm,
And then
okay.
we can look which one the method is very nice for get the yield of essential oil, something like that. Something that I do in the my research group, doctor.
Okay. All right. Okay.
Mhm.
Next we have questions from Noor Sazwani Daud, our postgraduate PhD
Yes.
student, similar with Mir Mashudin earlier. So, Doctor, what is the biggest challenge in extracting bioactive compounds?
Yeah. Very good question. The challenge is mean how we start to extraction,
Mm,
because when we are wrong using the method, it mean we cannot get our target.
okay.
For example, when you try to isolate some compound in halia,
Mm,
the first thing you should avoid wax and fat, right? Because this impurity, I mean, I call impurity or matrix, is very difficult when going to column chromatography. I
mm,
mean, I don't want to hard work. I want to have a strategy.
yeah.
So what I should do when I extraction halia, when the halia already in raw material, I using hexane.
Okay.
Why? Yeah, hexane. Because why? I would like to try avoid the wax and fat. It means fat and wax will be dissolved in hexane, right?
Mm,
Yeah. After that, I using water bath and then shake. It means all the material that have non-polar character will
mm,
dissolve in hexane, right?
mm,
Okay. And then I try to filtrate for after one night maceration, filtrate and 3 times, two 3 times. After that, I check the LC. What happened? The wax is don't did not appear again in TLC, in thin chromatography to check the fat. It means my extraction already avoid and throw away the fat and wax.
Okay.
It means my column, my process to chromatography is quite easy. Then I still have fat and wax, right?
Yeah.
And the second one, I using dichloromethane. So I gradually, doctor. Dichloromethane is can also throw away slowly and sometimes still can extraction our target. And the last I using ethyl acetate because sometimes ethyl acetate can also extract by like a compound they have glycoside, something like that.
Okay.
And the last one I using methanol and water. Why? Because sometimes in Halia they also produce sugar, right? Something like that. So my target in dichloromethane and ethyl acetate. Easy one. I didn't hard work column in the column chromatography because I already throw away the fat and wax, something like that, and also throw away the methanol and sugar and any derivative of sugar, something like that.
So to
Something
say
like
that in extractions of volatile compounds, choosing the appropriate extraction method
yes,
is
something like that.
important. Is the
Yes,
is the
something like
important
that.
parts so that you
Yes,
can get
something like
the
that.
kinds of
Yeah,
compounds that you want. All
yeah.
right. Okay. I think just to check again, Facebook, do we have any last questions? So if I could ask, maybe it was maybe the last question lah kot eh.
Okay.
So what do you think, Dr. Rico, that so we have this knowledge about ethnobotanical. Our community of indigenous people have used certain kind of plants, certain kinds of herbs for medicinal properties. But do you think that there are still um, some plants, some herbs out there in the forest that for some reasons that the indigenous people have not did not use them for medicinal properties, but actually they do have some medicinal properties. Maybe I don't know, like anti-dengue, malaria, or cancer, or whatnot. What do you think of that possibility?
Yes, doctor. This is a good question. Until today, based on my experience, if a doctor and have team for botany or something, some that we already some plant that we already know. Sometimes the local people atau suku asli, sometimes they didn't open all.
Okay.
So what we should do is mean we try to close with them. Sometimes actually this is social science, right? Work, social science work. But sometimes I go to the jungle with social science and I writing. And finally, I got something. Uh, I got something very important. It means all the people in the local people. Sometimes they didn't give us exactly something like that. They're using
Okay.
like recipe.
All right.
And then when the recipe, we can look what happened in recipe, something like that.
Mm.
This is some knowledge, doctor. I
So,
also encourage the doctor team, maybe if doctor go to some forest and they have local people, just writing what they have done with this plant, something like that.
so there are
Because some
still
plant,
some
yes, something
closely
like that.
guarded secret lah of their own.
Yes, okay. Macam tu, doctor.
Okay, all right. Okay, so I guess that's all for our
Yes.
webinar series. All the questions that we have today. But before we end, I just would like to check with our director over here, Professor Dr. Hisham Al Anshasi. Are you still with us?
Yeah, Dr. Nazrin. Assalamu alaikum.
Well, Waalaikumsalam, Prof.
Waalaikumsalam
I think your voice is a little bit low. If you can tune up maybe the volume.
Can you hear me?
Yes.
Yes. Can
Okay,
Prof.
excellent. Thank you very much, Dr. Nazrin, and special thanks for Dr. Rico Ramadan. It was great pleasure and honor for having you today in our second webinar in ICA. And I'm happy also to see a lot of your activities, which is already matched, especially when you see about biomass in cosmetics. I have also book chapter there with Prof. Widya. And I think Dr. Nazrin also have. And I hope This was very nice session, and thank you very much
Welcome, Prof.
for the information you shared with many people around the globe in our network in ICA. And inshallah, we are looking to welcome you in person in ICA
Yes, yes,
soon,
Professor.
inshallah.
Inshallah, inshallah.
Very soon.
Thank
Next
you
year,
very
Prof.
much. What?
Next year, inshallah, next year.
Inshallah, next year.
We
Inshallah,
already have a
inshallah.
plan. Yeah.
Yes.
Inshallah. I mean, thank you very much, Dr. Nazrin. Thank you, Dr. Lisa, and the team in ICA for organizing this seminar. And I would like just to close to say, Alhamdulillah Rabbil Alameen. And thank you very much. And see you soon, inshallah.
Inshallah, inshallah.
Assalamu alaikum wa rahmatullahi wa barakatuh.
Walaikum salam wa
Waalaikumsalam.
barakatuh.
Thank you very much, Prof. Hisham. So just to share with everyone, Prof. Hisham is live with us today from Alexandria in Egypt. So very busy director indeed. Okay, so just again to close up, I would like to say thank you very much to everyone from wherever you are around the globe for spending your time to join with us in a knowledge sharing session organized by ICA, and ICA stands for Innovation Center in Agri-Technology for Advanced Bioprocessing. So it focuses on the research related to food products and agri-technology, and we are based at UTM, University Technology Malaysia, Pago campus in Johor, Malaysia. So if you have any collaboration opportunities that you would like to get in touch, you can reach out to directly to our website, ICA. You can Google ICA, or you can even follow our Facebook page, UTM ICA Pago, for upcoming webinar series, or to also keep track with whatever programs, research activities that are being conducted at ICA UTM. And also, if you have any problems and or even opportunities to collaborate with our special guest lecturer today, Dr. Rico Ramadhan, I believe you can reach him out. You can Google Dr. Rico Ramadhan, and I believe he will be more than happy to answer your emails. All right, that's all. Thank you very much, everyone, and I wish you a good rest of the day. Thank you very much.
Thank you very much.
Thank you.
Thank you.
Thank you, Dr. Rico. Bye-bye. Hi,
Yeah,
Baisha.
bye. Bye. Thank you.
Bye.